In this video, we will be introducing the adrenaline signaling pathway and G-protein coupled receptors. I'm Jeslyn.
And I'm Emily. Have you ever been face to face with a bear? It is a pretty stressful situation. In such situations, the adrenal gland secretes the hormone adrenaline, which then binds to G protein coupled receptors.
GPCRs are an important group of proteins that play a pivotal role in transmitting signals within cells. In this video, we will explore the characteristics, functions, advantages, and disadvantages of GPCR, and then focus on a specific receptor, the adrenaline signaling pathway, to identify its key players, sequence of events, and the intended response. By the end, we'll have explained what happens in your body when you run into a bear.
In this specific pathway, adrenaline is the signaling molecule, also known as a ligand. Meet adrenaline, a hormone also referred to as epinephrine. It's hydrophilic, which prevents it from passing through the cell membrane, which means it meets the GPCR on the surface of the cell.
Now let's meet G-protein coupled receptors. GPCRs are a class of cell surface receptors that are primarily hydrophobic. These receptors are embedded in the plasma membrane of cells, where they mediate most cellular responses to signaling molecules such as hormones and neurotransmitters, as well as being responsible for other functions such as vision, olfaction, and taste.
So, what is the purpose of GPCRs? These receptors are incredibly versatile and participate in a wide range of cellular processes. They also help regulate many bodily functions by influencing broad physiological processes such as neurotransmission, cellular metabolism, secretion, cell growth, and immune response. They are typically used for sensing and responding to various signaling molecules. GPCRs can initiate diverse cellular responses, including changes in gene expression, enzyme activity, and cell membrane potential. They mediate the majority of cell responses to external stimuli.
GPCRs offer several advantages, such as their ability to amplify signals. One receptor is able to activate more than one G protein complex, and the effector protein activated by the G protein can create many secondary messengers. The activated protein kinases can each phosphorylate multiple cellular proteins, effectively amplifying a small signal into a larger response.
However, GPCRs can be overstimulated, and as a result of the body's natural mechanisms to prevent that, desensitization could occur. Overstimulation of GPCRs could result in cellular toxicity or uncontrolled cell growth. When limiting GPCR signaling, desensitization could occur, which is a decrease in response to repeated or continuous stimulation. If desensitization occurs, the receptor would require stronger stimulus to have the same response as before.
Mutations in GPCR can lead to side effects such as retinitis pigmentosa, hypo and hyperthyroidism, nephrogenic diabetes insipidus, and others.
Now let's understand how GPCRs work. When a signaling molecule, which is typically hydrophilic, binds to the extracellular domain of GPCR, it induces a conformational change in the receptor.
The guanosine diphosphate, also known as GDP, attached to the heterotrimeric is replaced by guanosine triphosphate, GTP, following the binding of an extracellular signal to the GPCR.
When GTP is bound to the G protein, the alpha subunit dissociates from the beta-gamma complex and activates the G protein. These changes activate an intracellular signaling cascade, ultimately leading to a cellular response.
The players involved in causing this response is the signal adrenaline, also known as epinephrine, the adrenic receptor, which is the GPCR that responds to the signals, the G protein, adenylyl cyclase, ATP, cyclic AMP, protein kinase A, abbreviated as pKa, and phosphodiesterase.
First, we have the signal adrenaline. When adrenaline binds to the adrenogenic receptor, it activates the G protein, which in turn stimulates the membrane-bound enzyme, adenylyl cyclase.
Adenylyl cyclase catalyzes the conversion of adenyl adenosine triphosphate, which is commonly known as the source of energy for many essential processes in cells, into cyclic AMP, abbreviated as CAMP.
CAMP is a second messenger that activates protein kinase A or PKA, which then phosphorylates various target proteins and amplifies the signal.
This process creates a response. Some of the major reactions to this response is to increase heart rate, dilate air passages, and redirect blood vessels to major muscle groups, including the heart and lungs. These reactions prepare the body for a rapid response to stress or danger, such as fight or flight.
The signaling pathway is terminated through the hydrolysis of CAMP into AMP by the enzyme phosphodiesterase. This deactivates PKA, stopping the signaling cascade.
To conclude this video, we learned that GPCRs are versatile receptors that play a crucial role in cellular signaling, including the adrenaline signaling pathway, which helps us respond to stress and danger effectively with the well-known fight or flight response. We hope you found this video helpful. Thank you for watching.
And here are the sources that we used.